Define replication, mention 4 inhibitors of replication. So the process of copying the DNA is known as DNA replication. So in the daughter cell, one strand is derived from the mother cell and the other strand is newly synthesized. What are the salient features of replication? Each strand acts as a template over which new complementary strand is synthesized, and it follows the base pairing rule. That is, adenine it pairs with thymine and guanine it pairs with cytosine. Then polymerization of the new strand, it is taking place from 5' to 3' direction. And the summary of DNA replication. So first, origin of replication is identified, then unwinding of the parental strand occurs. That forms the replication fork. Then RNA primer, which is complementary to the DNA template, is synthesized by RNA primase enzyme. DNA synthesis is continuous in the leading strand by DNA polymerase, that is towards the replication fork. And the synthesis, it is discontinuous in the lagging strand, that is away from the replication fork, and this is known as Okazaki fragments. Then in both strands, the synthesis is from 5' to 3' direction. Then the RNA pieces are removed. The gaps will be filled by deoxynucleotide by DNA polymerase. And then these pieces, they are ligated by DNA ligase enzyme. Then the proofreading process, it is done by another enzyme, DNA polymerase. So the main enzymes involved in replication are DNA polymerase, helicase, topoisomerase, DNA primase, single-stranded binding protein and DNA ligase. Example for inhibitors of DNA replication. Certain compounds, they will inhibit bacterial enzyme but will not affect the human cells. Such drugs, it is used as antibacterial agents. Examples are ciprofloxacin, nalidixic acid, then novobiacin. These are antibacterial agents. Then there are some other compounds which will inhibit human enzyme and they will arrest new DNA synthesis, and therefore they arrest the cell division. Such drugs, they are useful as anti-cancer agents. Examples are 6-mercaptopurine, 5-fluorouracil. Explain the DNA repair mechanism. DNA repair, it refers to a collection of processes by which a cell identifies and corrects damage to the DNA molecule that encodes its genome. So the DNA repair ability of a cell is important for the maintenance of integrity of its genome and for the normal functioning of that organism. So the different DNA repair mechanisms include excision repair, that can be base excision repair or nucleotide excision repair, then mismatch excision repair, then recombination or post replication repair. The general mechanism of DNA repair is the newly synthesized strand, it will not have methylated bases. So the enzymes, they can easily identify the original DNA template strand. Then the mismatched base, it is identified and removed with a few base around that area. Then DNA polymerase enzyme will synthesize a small segment of DNA with correct base sequence. Then that gap will be sealed by DNA ligase. Nucleotide excision repair, it occurs along with the replication process. So the original template of DNA will contain methylated residues and the newly synthesized strand will not have methylated bases. So the enzymes, they can recognize the original DNA strand. Then the mismatched base, it is identified and it is removed along with few bases around that area. In base excision repair, DNA glycosylase, it will recognize a damaged base and cleave between the base and deoxyribose. Then another enzyme, endonuclease, it will cleave the phosphodiester backbone near the AP site. DNA polymerase, it will initiate repair synthesis from free 3' hydroxyl at the neck, removing a portion of the damaged strand and replacing it with undamaged DNA. Then the nick remaining after DNA polymerase 1, it is sealed by DNA ligase enzyme. In mismatch excision repair, methyl directed mismatch repair recognize the mismatched base pairs. Then that will cut that mismatched base pair. Then the incorrect bases are removed. Then it carries out the repair synthesis.